Hi. Have you been seeing science in the news and in politics and just wish that you understood what scientists do for their job and why they make the recommendations that they do and really just how science works? I'm Dr. Sarah Margolin, and I have a PhD in cognitive and sensory processes, and I've been doing human subjects research and teaching classes in research methods for almost 2 decades. Welcome to my series, In Plain Science, where I show you just how science works. In this first episode, we're just going to talk about the general process of science, how we get from idea to recommendation. So the first thing that we do is we review the literature or what other scientists have done. And this is going to give us a sense of what we already know about that topic and what next question we should ask. That's actually a very hard leap to make and takes years and years of training to be able to think logically about what that next step and next question is. Once we have our question, then we can start to think about what kinds of methods we might want to use to answer that question. And the method that we use is going to be dependent on what we already know, what techniques might be beneficial in this area, and what techniques we've been trained on, what equipment we have available to us, and what is ethically able to be accomplished. And we'll talk about ethics in another episode. Once we've settled on a potential methodology that we think could answer our question, we're going to write up a big proposal. And that proposal is going to go to an institutional review board, or an IRB. And that IRB is going to review that protocol and that proposal for all kinds of things. Is it ethical? Is it maintaining integrity to the data? Is it maintaining anonymity and confidentiality? And these are all things that we'll talk more about in episode on ethics. And it's also going to look at, is this the most efficient or best way to answer that research question? Once the IRB has approved our methodology, then we can start collecting data. And that data collection is going to look like all kinds of things, depending on what our research question and methodology are. So data collection could look like asking participants some questions about their feelings on a particular topic. It could be running people through an MRI machine. It could be asking people to do memory tasks. That's something that I do in my lab, and where they have to remember items or read a passage and then answer questions about what they read. It could involve animals, depending on what our methodology is, in which case there's extra layers of approval from an IRB type board. And it really depends. Once we start collecting data, we're going to do it the same way over and over and over again until we get enough information to move to the next step. And the next step is going to be data analysis, where we now look at that data in big group averages to see what happened. So when we look at that data, we are usually comparing 2 different or 3 different or 4 different conditions. So this is where you might hear things referred to as like the control group or the placebo group, and how that might compare to a treatment or an experimental group. And we'll talk more about that in another episode. But what we're doing is looking to see if any differences between one group and another, or one condition and another, are happening because that's what happened, or most likely what happened, or if it's just fluctuations due to chance. When we get that information and when we get that answer, it's usually through statistical analysis. And that statistical analysis is going to tell us if this is the way it typically would work, or if this is happening by chance. Once we get that information, then we can start to draw conclusions based on those statistical analyses. And so those conclusions that we draw are going to be dependent on the constraints that we had in that experiment and the constraints that we had in the sample or the people that we tested, because we are sampling from a population, from a larger group. We're not using every single person that exists. There are going to be some limitations in what kind of conclusions we can draw. For example, if we're testing the efficacy or the effectiveness of a medication on women aged 20 to 40, then what we learned about that medication really only applies to women aged 20 to 40 and can't be extended to people and women who are postmenopausal. Importantly, the conclusions that we draw are based on large group averages. They are not based on individual people's data or individual anecdotes or stories about someone's specific experience. So when we say a medication is effective, we mean that it's most of the time effective. We don't necessarily mean that it's going to work for you, because we haven't studied you specifically and you're a unique individual. Now, once we've drawn those conclusions, you'd think we'd be done, but we are not. Because now we need to share our findings with the scientific community. And this can happen in a couple of ways. It can happen at a scientific conference. It can happen in a manuscript that's published in a scientific journal, which that's a whole process in and of itself we'll get to in another episode. There's lots of ways to share that information. But the reason we share that information is so that we can make recommendations, that we can have other scientists look at our work and ask more questions. We can have other scientists look at our work and try and replicate our findings to make sure that it happens that way again, right? And that we weren't just a fluke. And so the process then keeps going and going and going so we can ask more questions and get more answers. So if you liked this and you'd like to learn more about science, please make sure you're following along for the next episode of In Plain Science.